Thank you very much for giving me the opportunity to present these data and to also to present the company, which is in the field of protein production in eukaryotic cell-free systems. Now, the company is located in the southwestern part of Berlin. It's in the so-called FU hub, which is on the FU area close to the Freie Universität Berlin. This upper left U-shaped building, this is where the company B4 PharmaTech is now situated. Together with the Freie Universität Berlin, here we're collaborating with Professor Rainer Haag from the Department of Chemistry. The whole area is a brand new area with more than 66,000 square meters for startups, which will be available hopefully next year. Now, what is B4 PharmaTech doing? B4 PharmaTech is producing cell-free systems based on prokaryotic, but mainly eukaryotic cells. We do a fermentation step in suspension culture. We do centrifugation steps to collect those eukaryotic cells, namely SF21, CHO cells, HEX cells, K562 cells, and further cell lines, which are highly enriched by vesicles and which harbor the opportunity to produce glycosylated proteins also on cell-free systems. The cell pellet is then disrupted and purified by size exclusion chromatography. We replenish the system with an energy regeneration system, and those so-called lysates can be stored for more than 10 years, thereby avoiding any batch-to-batch variations. You always produce the protein of interest in the same batch. This system can be operated in a fully automated mode. On the upper left side and on the lower left side, you see the robot pipetting the cell-free protein reaction in a 96 well plate. The gripper is taking the plate, putting it into the incubation oven, and after 60 minutes, the maximum yield of protein is produced in this batch-based system. Now we're working with partners like such as Sinova. Sinova is producing these dialysis-based systems to improve the yield of cell-free produced proteins. So in this way, we're able to produce 1 gram per liter of high molecular weight membrane proteins, for example, in eukaryotic cell-free systems. This has been published by Lena Thoring. And here is a comparison, the confocal laser scanning microscopy, which shows that the CECF system, which is a continuous exchange cell-free dialysis-based system, leads to a high yield of membrane-embedded transmembrane protein. So this is another interesting option of this eukaryotic cell-free system. We have intact vesicles in the system with an active translocon, so also multimeric membrane proteins can be produced in this cell-free system. Here's one example, which was analyzed by Srushan Dondapati and also published. This is the thermostable direct hemolysin, which is a tetramer, and it forms a tetramer in the cell-free system, can then directly be pipetted onto electrophysiology devices and analyzed for its functionality, which is documented right here. I won't go into the details. Another company we're collaborating with is the company Cybertron, also located here in Berlin. Cybertron produces these small robotic workstations. So here we can produce proteins in cell-free system with within 1 square meter, where hundreds of proteins can be produced in parallel in these eukaryotic cell-free systems. Now, the system is also a system of choice for the introduction of mutations, which means also the introduction of non-canonical amino acids for click chemistry. Acid and alkene groups can be introduced into the protein. Toxic proteins that cannot be produced in any in vivo system can be produced in cell-free systems. And I mentioned already the synthesis of membrane proteins, but also the synthesis of antibodies, antibody-drug conjugates, clickable antibodies, single chains, nanobodies, and so forth are in the scope of our company. Now, with an artificial intelligence-based company, we do this kind of protein design in cell-free systems. After DNA synthesis, the protein is produced on the robot, and the functional assay determines the maximum yield of the protein designed by artificial intelligence. So the next step is an iterative way. In this way, we produce tailor-made enzymes for industrial applications. Also, those enzymes which cannot be produced in cells like proteases, DNases, RNases, where we have some special effects in cell-free systems to determine highly active enzymes. Now, this is the pipeline for the antibody-drug conjugate production in cell-free systems. We produce different antibody formats. We introduce a non-canonical amino acid, which is clickable, mainly para-acido-L-phenylalanine. By kappa-free click chemistry, we add linkers plus payload, and as we're working in a eukaryotic cell-free system, this can be directly pipetted onto the cell culture for further functional analysis of a novel antibody-drug conjugate. Our partner in the field of linkers is the company Iris Biotech, which is also a shareholder of our company. They produce different types of linkers, which can be analyzed in the cell-free system. So the business model for our company is antibody drug production, fine chemicals, diagnostics, also the analysis of mRNA-based vaccines in the cell-free systems, and this type of Amazon for proteins. So feel free to order any difficult to express protein. So the business model is shown here. We are now in the phase of a novel investment into kind of fermenters, machines, robots, and in the end there will be phase 1, phase 2 studies, hopefully with the cell-free produced antibodies analyzed in the cell culture. Thank you very much. I'm looking forward for partners for research and development. Investors are always welcome, and feel free to contact me for your difficult to express proteins. Thank you very much.